We're going to run through the things that you need to know for our first lab exam. And so the first 3 chapters that are listed under your lab exam have kind of minimal amount of information on them that you need to know. But for page 4 from your lab book, you have the directional terms. You need to be able to fill in the blank and draw on an image of those structures that are there for the directional terms. On page 5, you have the body planes, and then on page 18, you have the body cavities. So again, for each of those, you need to be able to label the drawings that are there. And then for page 20, you have the abdominal pelvic quadrants and regions. And so again, you'll have to be able to know where those quadrants and regions are within the abdomen and pelvis. And then for exercise 6, you have the histology component. And so for the histology side of things, I'm just going to run through kind of the characteristics that we can see on different slides of the different tissues themselves. And so here we have simple squamous epithelium, sometimes called squamous. And so these ones, remember, the cells are a single layer because it's simple. And so we're going to look and we're only going to find a single layer of cells. We're then going to look at the cell shape and we're going to see that they are flattened out cells. And so when we look to each individual cell themselves, they are flattened out here. And so this is a sample from a lung. And so these are the alveoli inside the lung, and we can see the individual simple squamous cells that we find inside there. And so that basement membrane tells us that that's the beginning of the tissue, and we then have one single cell, and then we come to emptiness. And so remember that for epithelium, it always ends in nothingness. And so there's always a border where we end up going out into an empty space rather than having tissue right next to it. And so we see that there. Again, the cells themselves are cuboidal. So under the microscope, they're going to look essentially like a square or maybe a little bit of a rectangle. But basically same height, same width for the cell itself with a nice centrally located nucleus. Simple columnar epithelium. Now the cells are taller than they are wide. The nucleus tends to be down towards the basement membrane there. And with simple columnar epithelium, this particular one that we have here is non-ciliated. So there's no cilia on those cells, but we may have ciliated varieties as well, which are going to have, switch over to purple here, we're going to have like little fingers that actually stick up from the cells that are there. And so all of those little ciliated components are there, and that's going to change the location for the tissues themselves. And so it's going to change where we find the types of tissues that we have within the body itself. And so the location is going to change based upon whether we have simple or whether we have ciliated or whether we have non-ciliated varieties. Next up, we have a ciliated tissue here, and so we can see that at the top of those cells, we have nice cilia sitting over the top of there. This one I have abbreviated as PSCCE for pseudostratified ciliated columnar epithelium. Big long name, but the tissue itself is not necessarily all that big. It's pseudostratified because the cells don't all make it out to the surface. So they don't all go out to the apical surface. Some of the cells are going to be shorter, some of them are going to be taller, and we're not going to have a nice flat top to all of the cells. And so that varies depending upon the cell itself that's there. With pseudostratified ciliated columnar epithelium, one of the other things that we find is goblet cells. And so we'll find these big goblet cells associated with them as well. Sometimes on this particular slide, it shows up as being kind of these dark purple granular type of structures. On other slides, it'll show up as being almost like a clear cell for those. Pseudostratified columnar epithelium also comes in a non-ciliated variety as well. And so it would look basically the same, except for no cilia at the top. Here we have stratified squamous epithelium. And with stratified squamous epithelium, we have the tissue once again beginning down here at the basement membrane. And in this case, the tissue itself is going to grow up from there. And so we're going to continue to grow out and out and out and out. We've got multiple layers, so this tells us that it's stratified. Down towards the basal surface, if we look at the cells, and especially in this region here, we can start to see the cells themselves here. They're more almost like cuboidal. And so the cells begin as almost like a cuboidal type of a cell, and then as they work their way out towards the surface, they become flattened out. You can see how they get more and more and more flat as they work their way out towards the surface, and those cells now take up that more squamous-like appearance. Stratified squamous in this particular case is a non-keratinized variety. We also have a keratinized variety, in which basically what we saw on the last slide was everything from here up to here. But with keratinized stratified squamous epithelium, there's an additional layer of cells which are actually dead. And so the last one was from the oral cavity, and this one is from the skin. Stratified cuboidal epithelium, we have now 2 cell layers. So 1 cell layer here, and the dividing line there is, the line that I drew there is basically the dividing line between the first layer of cells and the second layer of cells. And so stratified cuboidal is relatively rare within the body. We don't always find it, and even when we think we should be able to find it potentially, it's not always there in some of the glandular ducts that we would normally expect to find it. From there we have transitional epithelium. And so transitional epithelium, it appears to be stratified. We've got multiple layers of cells there, and it truly is stratified. But the thing is, with this, we don't really see a single cell type. And so there we have a more cuboidal looking cell, another one there. Here we have cells that are more flattened out. And then we also have cells that are columnar. And so transitional epithelium exhibits all 3 cell types. And what it allows for it to do is to go from this beginning thickness to then shrink down and stretch out as it loses its thickness and it gets stretched out, which is where we find it in places like the urinary bladder or the ureters. Moving into connective tissues, we're going to look, and one of the first things that we're going to look for are fibers. And so with connective tissues, we think of fibers rather than cells. And with those fibers, no matter what kind of connective tissue we're talking about, it began as embryonic connective tissue. And so it began as mesenchyme. And here we don't really have anything to note, really. There are some developing fibers inside. We've got lots of cells inside there. Lots of empty space in terms of the matrix that's inside there as well. And this is just the beginning stage. From here, it's going to differentiate into all our other connective tissues. Almost all of our connective tissues have visible fibers under the microscope, but there are a couple that don't. Adipose is one of them. Adipose, not only does it not have any visible fibers, but it's also one of those ones that really, once you look at it a few times, it becomes very evident as to what it is, because of these large vacuoles that each of the cells has. And so inside there are the lipids that those cells contain. Sometimes we see the nucleus of that cell, so that's the nucleus for this cell here. But depending upon how the slice was done, like these other ones over here, we may not actually see the nucleus for those individual cells just because of the way that it was cut. But it really looks like all of these big bubbles. This is different, however, from the almost bubble-like appearance that we saw back with stratified squamous. And so one of the big hallmarks in terms of the difference is that the edge here of our adipocyte, the vacuole that's inside there, the edge is nice and smooth because it's filled with liquid. And so it's filled with the lipids that are inside there of that cell itself. And so that smooths out the surfaces, versus on stratified squamous, it was kind of bumpy in a sense as far as the edges go. Now we have a connective tissue that has fibers. And not only does it have fibers, but it also technically has all 3. And so now we can start to see here, we have an elastic fiber. Here's another elastic fiber. And so elastic fibers tend to stain really, really dark. They tend to be nice and uniform in their diameter. So they have a uniform diameter along their length. They oftentimes don't really have very many visible branches that can be seen with them. And so we don't really see those there either. They're usually fairly thin versus collagen fibers, which are thicker, more robust. They don't stain quite as dark within the tissue itself. And then we can also sometimes see reticular fibers here as well. So areolar connective tissue is unique because we're going to see more than one fiber type. With blood, we go back to another one that does not have any fibers visible. It does have a large amount of matrix. And so all of the empty space here is all of the matrix. This is the plasma for the blood. And then we have all the individual cells. And so we've got red blood cells inside there. But for our purposes for right now, you don't need to be able to identify the blood cells. So you don't need to be able to identify any of the formed elements. You only need to be able to recognize that this is blood. And again, this is one of those ones that after you look at it a few times, it usually jumps right out at you. Osseous or bone tissue, we have something that always reminds me of a tree trunk. And so when we look at the structures here, we have a central osteon, which we'll talk about when we get into the bones. But then all of these lamellae, concentric circles around, we have something that ends up looking like a tree trunk almost inside there. But again, what you need to be able to recognize is that this is osseous or bone tissue. We're starting to get into connective tissues where there's not really a whole lot of space in between the fibers anymore. And so we're into dense connective tissues. And so dense irregular connective tissue, we've got all kinds of fibers going in all kinds of different directions. And so they're not parallel to one another. They're crisscrossing one another and things like that when we see those fibers. Dense irregular connective tissue is strong in multiple directions. And so with dense irregular connective tissue here, this is almost like if we took spaghetti, cooked it, put it in the pot, and then dumped all the water out, and then let it dry again. Now, instead of having kind of strength on either end and we can kind of pull it, now we can pull this in multiple directions and it's going to have some strength to it. Versus its counterpart, dense regular connective tissue, here all the fibers are parallel to one another. They're all going in the same directions. So although there's that little wave in the tissue there, that doesn't change the fact that they're all running parallel to one another. Dense regular connective tissue is more like the uncooked spaghetti in the box. And so if we were to pull end to end on the spaghetti, it's not really going to break when we pull end to end. Or even if we were to push down from top towards bottom, it's going to be more difficult for it to break in that sense on those. Versus its counterpart in dense irregular, it's going to be not as strong, but it can be strong in multiple directions. Dense regular is more strong just in a linear sense. Elastic cartilage, going into our cartilages, has elastic fibers. And so all of these little fibers interspersed through here, going all kinds of different directions, all those fibers are elastic fibers. Very, very stretchy, very, very resilient. So all those dark purple spaces inside there are the elastic fibers themselves. And so we can stretch it, we can pull it, and it bounces right back to its normal position. So this is what we find in places like the ear and parts of the cartilage of the nose are made up of elastic cartilage. We also have elastic tissue, and elastic tissue is what we find in things like blood vessels. Fibrocartilage, we're back to having collagen fibers again, but these collagen fibers are kind of long and wispy. It almost reminds me of something like taking a cotton ball and kind of pulling it apart and shredding it. The fibers that are there are kind of all going in that same pattern. They're also kind of like in layers. And so we see them oftentimes in these layers inside there, almost like layers of an onion. And so with that, we have very good compressive strength. We can push on it and push on it, push on it, and it doesn't give way. And so that's why we use it in places like the intervertebral discs and the meniscus of the knee. The hardest of our cartilage is hyaline cartilage. And so hyaline cartilage has kind of an amorphous type of a matrix inside here. And so all this kind of purple stuff here is the matrix itself. There are some collagen fibers in there, but it is not visible generally under the microscope. This one kind of almost sometimes reminds me of like stained glass in terms of its appearance there. There are the cells inside lacunae, so they're inside these little pockets inside there. And so we have the ability to form all of that matrix inside. Hyaline cartilage is essentially as strong as bone, just about, and we utilize hyaline cartilage in places where we kind of want to replace bone, like in the costal cartilage of the rib cage, or at the ends of bones where we're going to have articulation so that it can protect the two ends of the bones and they're not rubbing directly against one another. And then we have reticular connective tissue. Reticular connective tissue has collagen fibers, and those collagen fibers branch. There's a lot of branching to them. So they're going off in all kinds of directions. When we were to look at it, if we were to just kind of take the collagen fibers apart and kind of lose everything else, we would have something that resembles kind of like a net. And so we have this big network of collagen fibers, and they're designed to stop stuff. And so they're designed to trap things as they're going through there. And so we utilize this where we want to filter. And so in places of the body like the spleen or the lymph nodes, we have reticular connective tissue. For our muscles, we have cardiac muscle. And with cardiac muscle, we have the characteristics similar to that of skeletal muscle. Cardiac muscle is striated. It has all of these little like ribs in it. It almost looks like a worm to me. But then it also has these darker bands. These are intercalated discs. And these intercalated discs are special connections between the cells where we have gap junctions. And if you remember back to cells, when we had gap junctions that allowed us to transfer substances from one cell to the next. In this case, it's going to be things like sodium, potassium, and calcium, going from one cell to the next. Cardiac muscle also has the ability to branch, and so it has the ability to not just go in a linear motion or direction, it's going to have branches coming off and things like that. And so that sets it apart from skeletal muscle, in which skeletal muscle is parallel to one another. Each of the muscle fibers are nice and parallel to one another. We still have all of the striations. So we still have that kind of worm appearance to the muscle fibers themselves that are there. It doesn't branch. There's no intercalated discs. And it also has multiple nuclei per cell. And so every single cell has more than one nucleus. And so those are what set the skeletal muscle apart from cardiac muscle. Smooth muscle is nothing like the other two. Smooth muscle is completely different. And so smooth muscle is more of a spindle shape. And so it's thick in the middle, thin on the ends, and then the next one's going to kind of fit together with it like so, like there, and they kind of overlap one another. And smooth muscle we find in big sheets in places like the digestive tract and in blood vessels, allowing us to control the size of the organ itself. And that's about it for kind of the differences that we can see within smooth muscle. There's not a whole lot to it. Last up, we have nervous tissue. And so nervous tissue has these big cells. So we've got big cell bodies here for our neurons. And those neurons then have processes that come out of them. Those processes are our axons and dendrites. That's how they're communicating with each other. All of these little dots that we have are all part of the nervous tissue as well, these are glial cells or supporting cells. This is what is helping the nervous cells, all of our neurons, essentially stay alive and stay healthy and everything for them. So they're an integral component, and they far outnumber the actual neurons that we have, but they're not actually sending out the signals to the cells themselves. So remember to be able to identify the name of the tissue, where you can find it, so what its location is, and then from there be able to also name some of the specific things that I pointed out on each of the cells as well. And then all of the content that comes from the first 2 exercises will be on there also.